what's cable? That one. If you got, you can go straight in your hole, can't you? As it were. Yeah? Get it? Stick it in there. Fantastic. Right, coming up now on the main stage is Mr. James Ball, who's going to make us realise we're all going to die. Is that about an accurate statement, my friend?
Not all of us.
Okay, so you're going to live forever. Scott's already dead.
Hashtag healthy.
And John McAfee lives on in our hearts.
John McAfee definitely does live on.
Yes. Are you good to go? Are you wired up? Oh, hang on a minute. I'm not going to touch you. There we go. Are you green? Say something. Talk to me, Goose.
Well, this is sensitive.
Yep. Okay. Well, without further ado, ladies and gentlemen, is your clicker working? Happy?
Yep. Give me a sec.
Sure.
There we go.
Excellent. Well, ladies and gentlemen, without further ado, please welcome James Ball.
Sorry, I've just been stunned into silence by John McAfee there. So, thank you for that, Sam. Right, Metavirus, co-opting DNA synthesis. I'm not talking here about genetic engineering, except when I am. So, I just want to start with a quick word about models. I am not a geneticist. I'm not an expert in genetic synthesis. During this talk, I'm going to be using a lot of models and analogies. Most models are useful, some models are sort of true. These ones hopefully are sort of true and useful. But just bear in mind, someone who is an expert in this stuff will know better. Annoyingly, I also discovered after choosing the title for the talk that the Metavirus is a name for a family of viruses. This has nothing whatsoever to do with them. So, please just ignore it. The idea is that this is about malware being able to write biological viruses. So, the basics, genetic editing, talking about modern ways to do it, we have, if I can get that to work, CRISPR-Cas9. That's the most recent way. It's popular, it's cheap, really cheap. You can buy a kit for about $15, mail order, next day delivery. And it's an easy to use technology. I use technology in a loose sense. It's not electronic, it doesn't involve devices. It is edited proteins, which can essentially cut, copy and paste synthesized genes or other genes into a genetic sequence at a chosen point. So it's a full editing kit for whichever genome you want it to be. Roughly speaking, what happens is it's derived from a bacteria. That bacteria lives along, has a happy life, gets infected by a virus, finds a way to beat that virus, uses these proteins to cut out a snippet of that virus's code, store it in its own genome as a library to recognize it in future, so that it can use the same defenses again. Those proteins can be extracted or synthesized and then used to do the same thing, basically at will. And this is the type of stuff where, for a bit of money, you could do it quite happily in your home. Now, handy analogy, it's exactly what classical antivirus programs did. Store a signature, recognize it, react to it, contain, deal with the threat. Now, within the laboratory, that is exactly what it is. It's editing genes. It's not completely error-free. Mutations creep in, but it is the most cost-effective, the most accurate way that we have of doing genetic manipulation as of today. There are other methods, they're more expensive. I'm sure tomorrow there will be new ones coming up. There's two out there called ZFNs and Talons. They're older ones, not as popular because they take more effort, cost more money, and aren't as accurate. So, budget biohacking. Here you are. This is a CRISPR-Cas9 kit. You can get it, mail order. They do next day delivery, so you can get that as well. And you can have it ordered to your home. It's got roughly enough in there for about 5 experiments. Edit your own 5 organisms. This one's designed to allow genetic modification of E. coli. So you can splice genes into an E. coli bacterium. You can, of course, use the same techniques to splice it into anything else. And that's a non-dangerous variant of E. coli, not the one you need to be worried about in food. I just found the laser pointer instead. Here we go. Now, gamers spot the difference here. Can anyone tell me the key difference between these 2 sets of code? I'm hoping it's fairly obvious.
One's black, one's white.
Good enough. One's computer code, one's genetic code. The similarity is that they are both executable code. For all intents and purposes, this is one of those models I mentioned earlier. Computer code, we know it's executable. It runs through a compiler, runs through the computer, carries out instructions, does stuff. Genetic code is executable. Runs through the machinery of the cell, gets executed, builds proteins, those proteins go off, they're daemons, they're services, they go off, they do things, they build a human body, they build an eye, they build a deadly virus. But they are really both executable code, and there's a key thing there. For anyone who does reverse engineering, you'll be aware that getting from pure assembler to what the code is actually doing is a really tough challenge. It's much easier when you're working at a higher level and you can read the code in C or Python or whatever you want to. If it's been compiled to that level, it's much, much harder to work out what it's doing. Shockingly, the same applies to genetic code, because looking at that, I have no idea what that even codes for. But geneticists are having the same problems in that they get sequences, and the only way to really find out what they do is execute them somewhere. And then they might do different things in different circumstances. So, same as executable code on a computer, different OS, different processors might carry out different instructions, might just not work, might break the thing, set fire to the hard drive, could be anything. Same thing with genetic. You put it in a different organism, could have a completely different function. It's really, really hard to predict what it's going to do. And the amount of code involved is surprising. I'm actually going to ask for guesses. Who thinks there is at least a terabyte of data in human genetic code? Okay? Gigabyte? No. There is roughly 750 megabytes of data in human genetic code. The compression is astounding. You've got functions in there which are reused for multiple purposes when they're invoked different ways. You basically have the worst hack job of code you could imagine, with no comments, using every trick to jog memory, to reuse processes that you could imagine, running in every cell in your body. Which is, you know, encouraging. One day we'll figure it out, I'm sure. There's another problem, which is again fundamental to genetic code. If you're running something on a computer, you might be running it as a user, or you might be running it at kernel level, might be running it as system. Biology doesn't really do that. If it's in the genetic sequence, it all runs with the same privileges. You do not have permission levels built into cells. If it's in, it's trusted, it will execute. So, when you get a virus, what's happening is it is injecting its own genetic code into that machinery. That is then executing. It's usually creating more copies of the virus and often has nasty side effects. It's why we use the term virus for computer viruses as well, and why it's not always entirely accurate. Distinct from malware, a virus requires a host process. retroviruses in particular have persistence. So a normal virus, when it edits a cell, when it hijacks a cell, it will spew out loads of copies, the cell dies, problem, you get a cold, you get ill, you get COVID, something nasty, but it's not too bad. A retrovirus makes a permanent change to the code of that cell which is persistent as it replicates. So this is why genetic editing allows you to theoretically do things like change eye colour, deal with genetic defects, all sorts of different things. Particularly if you catch it early, before the cells replicate too much, you can completely change an organism at a base level in a way that is persistent throughout that organism's life and through any descendants. Imagine if you could just say, I'm going to pop over to Dell, take one of their master computers and inject my own malware into it. Okay, so given all that, I think I've managed to just turn something off, lovely. Scott, your clicker broke things. There we go. Is there a danger? Well, we've got an audience all wearing masks, so you know that there's a danger to viruses. Also, how do I turn off the laser pointer before I accidentally blind someone? It's fine, I'll just aim it downwards and try not to move my hand too much. Is there a danger of this? I'm going to call that dead. Right. Well, 2017, a group of researchers, and this caused a bit of a storm at the time due to ethics considerations. Can't imagine why that might be. They published a research paper on synthesizing the horsepox virus. Horsepox virus related to smallpox, not a great thing. Same sort of data, same sort of virus. And the synthesized DNA is about 100 to 300 kilobytes of data. We're not talking huge stuff here. They took that synthesized horsepox DNA and edited it into a live synthetic cell. So they essentially took an extinct virus and brought it back to life. And it was potentially infectious. What they really demonstrated was, if you have a record of the code, not even the live organism, of something like smallpox, yes, it's expensive to do, it takes a bit of a challenge at the moment, but you can synthesize that, edit it into an organism, and you suddenly have infectious smallpox. If you don't think this is a great idea, I'd agree with you. As I said, there were some serious ethics concerns around it. It does sound like the next Resident Evil film starting up, or Planet of the Apes. Genuinely think that's possible. But yeah, they revived an extinct relative of smallpox. This is not something you should do. Certainly not something you should do for an academic paper. They put forward the argument for the research that it would allow extinct viruses to be studied, to be prevented, should they research. Issue here, as people raised, was if it was still extinct, you wouldn't need to prevent it. Just leave it dead. They were virologists, so they knew exactly what they're doing. Research took about 6 months, and came to a total cost of roughly $100,000. Amateurs in a university, not looking to actually weaponize something. Now, all of us here are interested in security. I have this whole thing that security is holistic. You cannot distinguish too much between cyber, info, bio, physical, anything else. So, biosecurity. We all know about containment, we know about segregation, we know about keeping information, keeping assets safe. The last death from smallpox? Can anyone give me a guess on when? No? 1978. In Birmingham, where it was effectively extinct. The last actual case, live wild case, was in Somalia in 1977. So a year later, the last smallpox victim dies. A 40-year-old woman called Janet Parker. The infection vector was never really established, but she was working in the building where samples were being studied. Not in the same lab, but within the building. The investigation didn't uncover any failings. No one knows what happened. It was believed that it may have got through the ventilation, but that doesn't explain everything. And this occurred in a lab environment where there were no failures found in containment. There were no issues found with health and safety. It was believed they were doing everything fine. And she was not even on the same floor as the sample. So this is the type of thing that errors in security can lead to if you're doing biosecurity. We all know how easy errors are to make in cyber. The professor who headed the smallpox laboratory was one of less than a dozen authorised to study it by the WHO, and a day after her death he committed suicide. So it is a truly tragic story. Now, moving on to something a bit lighter and more hopeful. GenScript up there, these are all companies that will make synthesized DNA for you and post it out to you. GenScript, their pricing starts out at 18 pence per base pair. Base pair is roughly a bit of information, so you know, kilobytes, you're talking a few $100, a few £100. And that's with a turnaround of a few weeks to get it delivered through the mail to your door. Many manufacturers of DNA, many of the people who do this, apply a blacklist. They apply a list of known harmful genetic sequences. So if you try to order one of those and it's on that list, they will not create it for you. Many of them don't do that at all because it takes a lot of work. There's a lot of false positives. So they just say, hang it, we'll print anything. Even from those who do, the amount of work it takes, you have to get virologists in to properly understand when you've got a false positive, or of course, when you've got a true positive. If you break up a sequence, bear in mind we can edit it back together however we want in an organism. So if you break up a sequence, you can obfuscate the code and get that sample sent out to you in 2 samples, stitch them together afterwards. It's too inconvenient to stop someone ordering the genetic sequence of a killer virus. Too expensive. So effectively, let's say you can do it. Now the plus side is, we've solved this problem sort of in cybersecurity. We're quite good at antivirus, so maybe we've got something to teach them there. There are people working to solve this, but all of the stuff around it is voluntary. It's entirely voluntary. Companies do not have to do it at all. Right, security. Back to security. This is going to seem irrelevant for a bit. For those of you who remember the days of fax machines, you will almost certainly remember spam coming through the fax machine. For the rest of you, well, maybe some more of you who remember printers before we all went paperless, you may remember printvertising, spam coming through your printers. People hijacking internet-connected printers and advertising things. This was an advert for a printvertising service, so you can just order it and pay per 1,000. It's lovely. Anything internet connected is a potential attack surface. We all know that. And anything that's a potential attack surface will be used to generate profit or cause harm. 3D printers. A few years ago, brand new, really expensive technology, no one had them. Now you can pick one up in spare parts, internet connected. Still quite popular. Print from metal, print from sugar. I do like the sugar printers. Pizza printers are out there, by the way. Many hackspaces have built their own pizza printer. Don't know why, it's cheaper to order, but it's fun, I guess. And of course, you've also got printers that will print human flesh for grafting or for replacement. So you can have human flesh printed onto a scaffold, which can then be surgically reattached to someone, and it will heal. It replaces a body part. There's the same sort of harm, the same sort of potential for spam in these as there is in any other printer. There's also things like the 3D printed guns, the open source ones that you can download from almost anywhere you want with 5 minutes of Google work, print at home and have a working, might be a stretch, but some sort of gun. Which conveniently leads me on to, for anyone who doesn't know XKCD, I highly recommend it. It has actually happened. 3D printers have been hijacked in order to print penises. This should surprise absolutely no one. 3D printers aren't too dangerous. They're dangerous if someone deliberately tries to use them to create something malicious. They're a nuisance if someone starts printing winged penises out of your 3D printer and you have to explain to the lab head what's going on. You might be wondering what this has to do with the whole genetic sequencing, DNA synthesis piece. But just before that, I'm going to do a quick shout out to a guy called Chris Roberts, who roughly a decade ago found a way to go into tractors, a whole set of tractors, and update their firmware to change the planting distance. If they had pushed that update out, it would have meant that every single automated tractor from that manufacturer planted its crops too close together to get a harvest. Given that this planting usually happens roughly around the same time, and this sort of attack might have been difficult to spot, and farmers don't order more than they need to in terms of planting, so they would only have the stocks they need, you would have then completely destroyed the grain harvest with a malicious software update. And that's not just in America, that was anywhere these were used. Now this is a lovely device. I really want one of these, so if anyone has some money to spare, you can buy me one. This is a desktop DNA synthesizer. I can take it and I can print whatever DNA sequence I want to. I need to get the right cartridges for it. They're amino acid cartridges, but you know they're fairly cheap, about $800 for one of those. And I've got no safety list there. Even the limited checking that the companies who do that synthesis have doesn't really apply. Even if they stick it in the device somewhere, that's a device I've got physical access to. There is nothing that's going to stop me doing what I want to with it. This costs 15,000 euros, this particular one. So, you know, bit expensive, bit more than I can afford right now, but 3D printers cost that just a few years ago. It has network connectivity, so you can connect it to the internet and print from the convenience of your own home, at the lab, or at the hospital, because these things are used for creating retroviral treatments. So, some hospitals have them, one's at the cutting edge of this research. Now, I don't really have a nice, clean, and here's the solution thing to end this on, or a twist that makes it all funny. We're just starting to recover from a pandemic which has, no matter what you think of number of deaths or anything else, severely negatively impacted the world. A few years ago, we might have called this unimaginable, except that it was predicted repeatedly. We knew exactly that this could happen. It has shown not only how ill-equipped we are as a world to deal with this sort of thing, to deal with a novel infectious virus, but it also comes at the moment when the means to manufacture a virus is available to anyone with the right expertise and 15,000 euros, or the right expertise and an internet connection which can get them into a hospital's one or a lab's one. This sounds like sci-fi, and I do genuinely hope it stays that way. But I just want to give you an absolute nightmare scenario. Someone malicious spends time, gets the expertise to create a custom-tailored virus, one targeting specific traits, one targeting a specific area. Myxomatosis was a genetically engineered virus. So they get the expertise to do that. They compromise a hospital's desktop synthesizer, which is being used to print retroviral treatments. The hospital carries out the procedure. You now have that new viral code dropped into someone in a hospital with possibly no one having any idea until it's too late. So when I said I really have no stinger, I don't. I've got no clear ending to this. I have just got pretty much dread. I researched this a while ago, and everything I read into it just made me feel worse. I don't even really have a call to action. The technology is out there. The people with the expertise to do something about it are trying to, and it is completely inadequate. We should all be familiar with that feeling. And this is genuinely a potential problem. People have synthesized a live, infectious, extinct virus. They have done it. They have done it and published the research on it. So it may not just be potential, it may be proximate, this may be close. I am very open to any ideas about how to sort this sort of thing. I don't mean to alarm, and I'm not saying we're all going to die, but I am saying it is a possibility. Now, during this, just to establish that I've got some idea what I was talking about, I did actually do far more reading than I wanted to. I had nightmares after this. There's the sources available if you want to check any of them yourselves. And I'd love to know if there are any questions or solutions. Solutions would be great.
Hang on.
Alright, so I work with an organization called Orbit RRI, which is Responsible Research and Innovation. And one of the things that we do is look at these types of things, including, for example, chemical printers, where it might be able to print off ibuprofen or bomb-making material. So do you think that, for example, although you might not have a call for action yet, that particular group should also start be looking at this type of stuff?
Very much so. There is a group out there, as I said, who are working on ways to solve this. It would definitely be worth looking at this sort of stuff and getting in contact with them, because their specialty is in genetics, so they understand it far better than I do. And they're very easy to find.
Cool, thank you.
So your genetic sort of like synthesis machine for $15,000, is there nothing there that, well, we have printers where if somebody sends a printer, a print-to printer that stays in the spooler, and you go to the printer and you use your like ID card to actually be scanned, and it says, yes, I've got one in my school for you, I will print it. If it remains in there for more than 15 minutes, it's dumped, just to make sure that it's going to the right person. It's not going to...
Yeah. There is potentially, yes, these printers currently take about 24 hours to finish a job, so it's not a 15-minute job.
Before it actually starts printing, it's in the...
Yeah, it will sit in a buffer before then, but those buffers are potentially vulnerable. If someone can get access to them, if someone can get access to the firmware on the printer, or there are any vulnerabilities. And I've not been able to find anything at all, and this was one of the worst bits, about any security testing done on these devices. Nothing. So if there's a vulnerability, any safety measure like that could be compromised. I would hope that they've built things into that to prevent it, but my specialty is security by design, and I know how poorly that's often done with things like simple mobile apps, which are really, really easy to do it for. So I do not trust that they have done this, and I've had no assurances otherwise.
Cool, thank you. Does anyone else have any more questions? Cool. Ooh. I will go this way. Thank you.
Couldn't the answer be just to not have these things on the network, so that you need physical access to them? That's the simplest...
Yep, it does. The same applies to printers, 3D printers, light bulbs. People put light bulbs on the network. Doorbells, dog feeders, cat feeders, keys, door access systems, cars. So yes, the simplest solution is to say, you know what, you have to do physical access and we'll apply physical security. I just don't think it's going to happen reliably.
Cool. Yep, I'll go this way.
Thank you. When you spoke about the potential attack, you sort of went quite quickly through obtain the knowledge, the genetic knowledge required.
Yes.
I've got a feeling that's not quite as easy as you...
No, no, it's not.
Is that barrier to entry enough? Because the motivation behind an attack would be one of terrorism, like anarchy. It's not. There's no way of profiting from it, I would imagine. So, is it that the barrier of entry is high enough to reduce this as being a kind of viable proposition?
Possibly, but I'm not going to say I'd rely on that, because while we can't see a profit motive in destabilisation, terrorism, the world as we have it today shows that plenty of people do. They see benefit to them in it. If a country can build a nuclear weapon, and a lot of them can, they can get the expertise to do this. It takes study, it takes training, it would take years of research and effort, but all of the information is available. There are people who theoretically could do this. They could then work alongside cybersecurity, unethical hackers in order to actually exploit that and apply that. So there is a barrier to entry, and it is a knowledge barrier. I'm not entirely comfortable relying on that.
Awesome. Anybody else have any questions while I'm here? Cool. Yep. Oh, you first.
Yeah, thank you. So, you know, you said there was some work ongoing. Is that all technical, or is there anyone doing anything like at a policy level to try and...
I'm not aware of anything being done at a policy level. And for those of you who made my think tank talk earlier, this would be a place to apply that and build our own think tank. But no, I've not found anything at a policy level. There is this effort by a group to do a better way of doing that checking, but they've not got very far, and it's got no legislative backing anywhere in the world at the moment. There are countries where this sort of research is illegal. I don't see that makes much of a difference.
Cool, thank you. One over there.
Yeah, hello. Sorry, it's just not so much a question, but just to address that. So So typically in the third year of a biology or biochemistry or virology degree, you end up doing a lot of the things that were discussed on this. So out of like a 4-year course where you have an integrated dissertation, you can have the people learning the skills in third year and then applying them actually in a lab in the 4th year. So a lot of undergraduates will have these skills. So yeah, just FYI.
Thank you, I feel so much better.
Yeah, thanks for terrifying us. Wow. Sure.
Surely the risk is from optimization of whatever the thing is, rather than creating something new, like COVID or whatever.
Yep.
And then you've got the Wuhan question of how that would have come about.
Bringing back smallpox would not be a challenge. Let's put it that way.
Mildly terrifying? Yes.
Just throw it.
Can you frisbee?
I'm thinking purely hypothetically here. You know how you can't print money? Would there be a way to sort of like, for every like viral sort of imprint, just kind of put like, you know, something that would stop it being printed?
So that's the whole idea, that printers have a device that recognises you're trying to print money and prevents it. The answer is yes, you could do something like that. You would need to somehow be able to recognise every potentially harmful genetic sequence, which could be every genetic sequence, so the false positive rate would render the technology useless. Even if you solve that problem, if I've got physical access to the device and can find out how you've applied that safeguard, there are ways around it. But it would be an obstacle. The more obstacles, the better.
I think you've sufficiently terrified everyone, which is usually how our conversations go.
This is why I don't sleep anymore.
I've noticed. Anyone got any more questions? Any final questions? No? Cool. Well, everybody give a warm round of applause for James.